Excellent. So yeah, starting out with this document, I just want to run through these 3 new stains. So far we've talked about 3 stains. We've talked about the negative stain, which is just staining the background, not the bacteria themselves. And then we have 2 different differential stains, our gram stain, which uses the crystal violet and the safranin, and then our acid-fast stain, which is testing for mycolic acids, and that one uses the carbonfusion and the methylene blue. So those are the 3 stains we've done so far, 1 that's not differential and 2 that are differential. These 3 stains remaining here are the next 3 that we're going to talk about. We're going to talk about capsule stains, we're going to talk about endospore stains, and we're going to talk about flagellar stains or Flagella stains. So starting with capsule stains, so similar to your acid-fast stain that was testing for a virulence factor, which would be the mycolic acids that are found in your Mycobacterium and your Nocardia genres. The capsule stain is also testing for a virulence factor for the capsule. Capsules are made of glycocalyx. That's the term you should probably know. And glycocalyx is kind of like a loosely bound slime layer that surrounds the cells. So scrolling down to the bottom of page 1, if you're following along, there's a couple of different pictures of a capsule stain, and let's talk about what we're seeing here. So seeing here on the right picture, you see there's kind of bubble-like features, and there's dark shapes in those bubbles. So the dark shapes in these bubbles are our bacillus bacteria, our rod-shaped bacteria, and then surrounding them in this bubble is the capsule. So essentially the capsule is this thick space around each of our individual bacteria. On the left-hand side here, it's a little bit further out. You can see the dark bacillus-shaped bacteria again, and around each of those bacteria, you notice this clearing. And it's not really a clearing, it's that loose capsule slime layer that surrounds the bacteria and protects it. It protects it from some of the things here that it talks about, phagocytosis, is why it would be considered a virulence factor, because it makes it hard for our bodies, hard for other animals to eat these bacteria, and so it kind of protects them and gives them an advantage. So capsules on individual bacteria are one thing. When you get a hard, it forms what's called a biofilm.
You're breaking up a bit.
Oh, am I? Ew, it says that. Network out and jump back in. So yeah, so the important thing would be to kind of learn, really understand, what a capsule would look like, so to be able to identify a picture whether or not a capsule is present. Wow, did that just kick me off again? Sorry, like they're doing something weird. So whether or not a picture has capsules or doesn't, so both of these would be positive for capsules or positive for glycocalyx. And then why this would be important in biofilm formation or plaque formation, and why it would be considered a virulence factor. So that's probably what these questions talk about there. The next stain we're going to talk about here is the endospore stain. So capsules are considered virulence factors. Endospores might also be considered a virulence factor. So endospores, what are they? They are formed when bacteria undergo stressful conditions. Some bacteria have the capacity to produce a spore phase, where they basically encase their DNA in a capsule and protect it from harsh environments. While it is encapsulated or while it is in the spore form, it's considered to be dormant. But once conditions get better again, these spores, like a fungus, these spores can germinate and they can grow back into bacteria. So it notes here some of the common genera that have spores are your Bacillus and your Clostridium, both pretty important for different diseases. The endospores help to prevent us from being able to kill them, and therefore they kind of give them an advantage in causing diseases. So the endospore, like the capsule stain, is looking at glycocalyx. The acid-fast stain is looking at mycolic acid. The endospore stain is looking at this dipyclonic acid complexed with calcium. So that's kind of what the endospore is made of, and it's protecting the DNA of the bacteria just as basically a shell surrounding the bacteria DNA. Are a couple of the diseases that you guys might be interested in caused by endospore forming bacteria. Bacillus, again, like I said, commonly produce endospores. Bacillus anthracis is what causes anthrax poisoning. So if you suspect anthrax to be present, you could test for the presence of spores, or if you have this on a slide from an infection from somebody, you could test to see if there's endospores present. That's a reason why it's fairly persistent and can be used as a bioweapon, because the sporulation form can be kept as a powder. You don't have to keep it in any sort of special conditions, because it's protecting the DNA of the bacteria. And then if you breathe in those spores, like they were being mailed around, if you breathe in those spores land in areas where there are better conditions, and those spores can germinate and cause an anthrax infection. Anthrax isn't something that's very common, but it is common in certain fields. For example, my husband is a wildlife biologist, and they have to be cautious of contracting anthrax from wildlife. It's also common in sheep. So if you're handling sheep, if you're doing wool shearing, those kind of things, it's also common in sheep wool. So just very different. If you have a patient with a background in some of those things, you might need to test for certain diseases, which is kind of cool. Another one they talk about here is Clostridium forming spores. Clostridium causes C. diff. You might have heard it called, but it's because of the name of the bacteria, Clostridium difficile. And this is what is caused after you take a lot of antibiotics. C. diff, because it isn't easily killed by antibiotics, it can colonize the gut and become a problem. So different diseases caused by endospores. So let's look at a couple endospore slide pictures. And from these pictures, you could potentially have a question asking whether or not endospores are present. So endospores are going to look like, let's see if we can zoom in on this a little bit. Endospores are going to look like little dots in the bacteria, or little dark areas. So here they have them stained in blue, but also there's ones that weren't stained that are these dark purple dots. So if you're looking at a bacterial slide that has a lot of like dark dots in the middle of the bacteria, that's probably indicative of endospores. And you can see this little cell over here doesn't have any dark dots in it, and that would be indicative of a vegetative cell, as it shows there. So this slide up here, you see that the endospores are stained in this greenish color. And you can see some of these cells don't have endospores, some of them do, and this could be because they've just been exposed to UV light or something like that, and some of the cells have gone like, oh no, and they started sporulating, and some of the cells are just like hanging out and gonna eventually die, or start forming spores eventually. So there can definitely be mixes on the slides. Let's zoom back out again. Oops. Okay, so definitely know what an endospore slide looks like positive and what one would look like negative. Know what endospores are made of, and describe why that would be maybe considered a virulence factor as well. The third type of stain we're going to talk about is a Flagellar stain. So bacteria have different appendages that are used for movement, and these are known as flagella. And there's different types of flagella. The reason why this is considered a specialty stain, like the previous two that we're talking about, is that it takes a little bit more finesse or a little bit different procedures to produce these stains, because either the features are very delicate, such as the flagella, or they're hard to differentiate, such as the endospores or the capsule stains. It's hard to stain the inside cell as well as get the picture of the outside of the cell as well. So these are a little bit more complicated stains, which is why we're not going through the procedures of any of them. But it's good to know what you're looking at when you're looking at these types of slides. So for the flagella, these features are so delicate and breakable that you can't really see them with a light microscope unless they're thickened. And they can be thickened by complexing dye with a mordant, very similar to your Gram stain, where you use the iodine as a mordant with the crystal violet, and they complex together. The dye used on the Flagella stain complexes, and it forms a thicker feature that you can view. So one of the important things would be to study here are the different types of flagella formations. So there's monotrichous, which is a single flagella on the end of the cell. Lophotrichous is several flagella on the end of the cell. Amphitrichous, one flagella on each end. And paratrichous, which is many flagella all around the cell. So let's see if we can identify a couple of those. This first picture here, if you're looking at the top of page 7, I think, you see that there's these thin thread-like features on the end of these cells. So this is going to be either monotrichous because it's sticking off the end, or amphitrichous. There's not many on one end, but we actually, if you look at this picture, you can't actually tell if they're sticking off both ends because you can't really tell where these bacteria end, or if they're only sticking off one end. So I would take an answer of amphitrichous or monotrichous for this particular picture. The second one is a little bit more interesting, and there's a couple reasons why. So these particular bacteria, the 4 on the bottom that you can see, with all of the flagella off to one side, you might think that this would mean that it was Lophotrichous. But if you know what's happening in this picture, you can tell that it's definitely what this little bacteria is doing up here, that top bacteria. It's actually a peritrichous cell. And the reason is these bacteria are actually using a process known as chemotaxis. So chemotaxis is how bacteria move towards food sources. They can sense the chemical signature of something, and by sensing that chemical signature, they either move towards it or away from it. So these bacteria, the bottom 4 bacteria, are all actually moving towards something. And this top bacteria here has tumbled, what's called tumbling. So bacteria either swim or tumble, and when they tumble, they spread out all of their flagella, and then they choose a random direction, and they'll start swimming in that random direction. And if that random direction is closer to the chemical signature, they'll keep swimming in that direction. If it starts to be more away from the chemical signature, they'll stop, they'll tumble again, they'll choose another random direction, and they'll go in that direction. And it's basically like a game of hot and cold. They'll just choose at random a direction, and if it's colder, they'll stop, and if it's hotter or warmer, they'll keep going until it gets colder, and then they'll stop and turn directions again. So this is actually a paratrichous cell, so there's flagella all around it, but once it starts moving in one direction, all of those flagella are flowing with the water, so it looks like they kind of gather on one side of the cell. Yeah. So again, just make sure that you can identify the different types of flagella, and maybe a couple of the details about why we would have to stain flagella differently under a light microscope. So those are our 3 specialty stains. And then in addition to those, for the practical, definitely want to know your gram stain, your acid-fast stain, and your negative stain. Okay, so let's next talk about the other documents in here. There's 2 different documents here. There's the bacterial growth curves, and there is the bacterial quantification. And I forget, they're both very similar to each other if you're just reading the documents, and one of them basically just has way more information than the other one. So we'll use that one, whichever one it is. So these are both the documents from the Labster manual, and definitely give yourself plenty of time to go through these Labster simulations. They're pretty intense, but they're also really full of some great information. So the learning objectives for these are to be setting up a serial dilution, and we're going to talk about what a serial dilution is. We're going to talk about how to calculate CFUs per milliliter, or colony forming units per milliliter, given a dilution factor and a volume, as well as the number of colonies. And then you guys are going to practice setting up these experiments and answering some questions as to why this is important within the simulation itself. So definitely read through the theory. It's very important for understanding generational times within bacteria. So bacteria multiply in an exponential fashion, which can be represented by 2 to the n. Because if you have one bacteria becomes 2 bacteria, 2 bacteria become 4 bacteria, 4 bacteria become 8 bacteria, and so on and so forth in an exponential fashion. So you can see here after 3 generations, it could be represented by 2 to the 3. You could basically have 2 times 2 times 2, or 8 cells. If you had 4 generations, 2 to the 4, it'd be 2 times 2 times 2 times 2, which would be 16 cells, and then 32 cells, and then 64 cells, etc., etc. Depending upon the species of bacteria you have, this generational time will differ. And then the next one they talk about here is colony forming units and serial dilutions, which I want to talk about a little bit here. And I don't have my pad set up at the moment. Grab that real quick, but I may post a separate video on some of this stuff as well, as if I assess that we're going to need to explain it a little bit more. So colony forming units, or CFUs, are what you're going to be commonly seeing as you do anything in microbiology. You're going to see CFUs, and it's usually shown in a way such as CFU per milliliter. So the colony forming units are just when you produce a plate of bacteria and you see all the little dots on that plate of bacteria, the colony forming units are the number of dots that are growing. So it is a representation of the number of live cells that are growing in that colony. So for the example that they give here, and we're going to walk through this on my Jamboard here in a second, but they represent the three things that we're looking for to calculate colony forming units, or the CFU per milliliter, is your number of colonies, the amount that you plated, and the dilution factor. So there's three different things that we're looking at. CFU per milliliter can be calculated essentially as the number of colonies divided by your amount that you plated, and then that can be multiplied by the dilution factor that you have, which is shown here in the serial dilution section. So let's talk about what this means in kind of more of a visual aspect. So this is what it's going to look like in that with you guys. It's on the document. Somebody's but yeah, so this is what it's going to look like. You guys are going to have a little computer to do these calculations, and you're going to little pointer here, have the number of colonies on your plate, and you're going to divide that by the volume that you're adding to the plate. So I'm going to add a new screen in here. So what does this mean? So when you have a plate of bacteria, you guys have your plates. You're going to take from your tube of bacteria a certain amount, and when you take that amount and add it to your plate, you're going to be adding a certain amount. So either you're going to be adding 1 milliliter, or you could be adding 0.1 microliters, or you could be adding 20 milliliters. So whatever this number is, let's just assume that we're going to go with actually 0.1 milliliters is what we will use a lot. So let's just assume we're going to be plating from our stock culture. So if this is our stock culture of bacteria, and we want to plate it to see how many bacteria there are, we're going to this amount. After we incubate this plate for 24 hours or 48 hours or something like that, it's going to end up with, you know, little dots all over it. That was really bad dots. Little dots. Okay. So whatever number of little dots are on here is going to be your number of colony forming units. So for example, if this is 25 colony forming units, and your amount that you use to plate is 0.1 milliliters, I'm going to break out my phone calculator here. I encourage you guys to do so as well. It would be 25 divided by 0.1, and that would equal 25 divided by 0.1 equals 250. So it would be 25. Let me see if I can write this in here. divided by 0.1 equals 250. I really wish I had my, and this would be CFU per milliliter, 250. So that is how you do this calculation without a dilution factor. So this is if your dilution factor was essentially, that's really annoying. If your dilution factor was essentially 10 to the 0. So this is what you would do to calculate your CFUs. You would take your number on your plate, the number of colonies that you grew, which would be 25. You would take the amount that you plated, which would be 0.1. You would divide those, and you would get 250 CFU per milliliter. So if we go back to our colony forming units here, that is what happens when you don't do a serial dilution. If you need to do a serial dilution, because you're working with so many bacteria that you can't count the bacteria, if you plated them on your plate, it would just overgrow the plate because there's millions and millions of them, and you can't count 25. You're going to need to do a serial dilution. So this gets into a little bit of the theory of the serial dilution, and you guys can read through this. M1 V1 equals M2 V2, so mass times volume equals mass times volume. And this is just how to, and if you walk through this, this is how to do the dilutions to obtain 10 milliliters of a 1.1 millimolar solution from a stock solution. It's kind of esoteric to what we're actually doing, but it does have a good point in that if you look down here, I think this is what my takeaway would be from this discussion here. Dilution factor, which we're going to be talking about, equals 1 divided by the final concentration over the initial concentration. And they have to be in the same units. And for this particular example, our final concentration is 1 millimolar, which is 0.001 molar. Our stock solution is 1 molar, so our dilution factor would be 1000, because essentially we are dividing 1 divided by 0.001 and dividing that by 1, which equals 1000. Yeah, so I wanted to talk to you guys a little bit away from this stock solution idea. I want to talk about serial dilution as it relates to actual bacteria on plates. So I'm gonna put another picture in here. So if we incubate a plate, oops, let's copy that, of bacteria, or a tube of bacteria. Let me draw my tubes here. In order to do a serial dilution, basically what we're going to do with our bacteria is have our stock solution. So this would be our stock solution. Would be 10 to the 0. Oops, that's really funny. And we want to be able to get it down to a area where we have between 30 to, well, that did not work, 30 to 300 CFUs on our plate. So if this stock solution has 1,000,000 cells in it, and we have to get down to 30 cells on our plate, we're going to need to dilute it serially. So we're going to have to set up a series of tubes. We're going to fill each of those tubes with 9 milliliters of a solution. And we're going to take 1 milliliter of our stock solution and add it into our first one. So this is going to be adding 1 part of our solution to 9 parts of our dilution. So we're doing basically a 1 to 10 dilution. So there's going to be 10 times less bacteria in this tube than in this tube, essentially. So in this case, our dilution factor now in this tube is going to be 10 raised to the 1, because we've just diluted our tube, and so our dilution factor is 10 to the 1. You would mix this tube up, and after you mixed it, you would then transfer sterilely 1 milliliter from this tube to this tube. And what we just did there was we made this tube 10 times less concentrated than this one, which would actually be 100 times less concentrated than the stock solution. So this would be 10 my raised to button. And you can probably guess what we'd do next. We would mix this one up. We would then pipette 1 milliliter from our 10 to the 2 into the 9 in our final tube here, and this would be a concentration or a dilution factor of 10 to the 3. So this particular tube is 10 times less concentrated than this tube, which is 10 times less concentrated. This one, 10 times less concentrated. This tube is 10 times less than this one, 100 times less than this one, and 1,000 times less concentrated than our stock solution. When you make these serial dilutions, you then plate each dilution. So if I plated onto this dilution, again, it would be that value that we were plating at, which would be 0.1 milliliters, potentially. I would take from the stock solution, and this plate would be our, basically our 10 to the 0 plate. I would take I should use a different color or something. I would take into this one, I would take an additional 0.1 milliliters onto this plate. And because this is our 10 to the 1 tube, this plate would be considered our 10 to the -1 plate, because it actually is 10 times less on this plate than is on this plate. And this is our dilution factor. So I should note that this is dilution factor, which is represented by like a positive number. So this would be 10. This would be, all right, so this would be 0. This would be 10. This would be 100. This would be 1,000. Whereas our plates here are represented by decimals. So this would be essentially 0. This would be 0.1. And then if you did this next tube over into here, the red one, this is getting very messy. I apologize. And we would then plate this onto this plate. So we'd have 1 plate for each of the tubes that we made. It'd also be 0.1 milliliters. And this would be our 10 10 to the minus 2. So, so each tube that we create through this serial dilution is going to have a factor of 10 less bacteria in it, and we can plate that to see that result in how our bacteria are growing. And so the important numbers that you would get out of here would be, I think I actually made a slide of this, would be your number of colonies that ended up growing on that plate. So for example, let's say we had 25 colonies growing on that plate. The dilution factor, so that's looking at your dilution factor of your tubes. This could be 10 to the 1, this could be 10 to the 2, could be 10 to the 3, depending upon which plate you're reading. So let's assume this is 10 to the 4 or 10 to the 3, since we only did 3 plates. So this would be 10 to the 3, which is the same thing as saying 1,000 times less concentrated than the original stock solution, and the amount plated. So that's, again, that amount that you're actually pipetting onto the plate, which in our case was 0.1 milliliters. So how does this work in actually calculating your CFU? So how do we calculate our CFU per milliliter of a serial dilution? So we saw that our CFU per milliliter at 10 to the 0, or essentially without a dilution, would just be 25 divided by 0.1. With a dilution factor, it would be, let me go out to our page here where it shows it. Actually, I didn't even show it here. You guys are going to be doing this quite a bit in the simulation, and I tried to and this is where I may pull out some additional pictures from the simulation and kind of create a separate little video. But basically what you're going to be doing here is you're going to be taking your number of colonies divided by the amount plated times the dilution factor. So if our plate is 10 to the 0, it would be 25 divided by 0.1, which would be 250 CFU per milliliter. But if we were looking at 25 colonies on our 10 to the 3 plate, this would be 250 CFU times 1,000. So it would actually be 250 times 1,000. Can't do math right now. That's what calculators are for. So that would actually be 250,000 250,000 colony forming units per milliliter in your original stock solution. So it would be this number. Let's see if I can type this out. So your number of colonies divided by your amount plated and then times by your dilution factor, which in this case would be 1,000. Wow. Computer not keeping up with me. So by your dilution factor, and that would be, in this case, this is going to equal 250,000 CFU per. So whether the number of colonies on the plate is informative. So you could look at a plate with 50 colonies and you could look at a plate with 25 colonies and you could think that the number 50 shows more growth. However, you have to take into account the dilution factor of that plate, because if it is 1,000 times diluted and you have 50 colonies versus 1 time diluted and you have 25 colonies, you're going to have, 2,000 bacteria versus 200,000 bacteria or 500,000 bacteria. So you could have different amounts of bacteria that are not representative of the number of bacteria on the plate. I know that can be very confusing. So I want you guys to, take plenty of time to work through the CFU calculations in the documents here, as well as in the simulation. And again, I will try to post something to clarify some of these components that we're running into that people have questions about on doing these CFU per milliliter calculations. A lot of the other theory in these documents where they're talking about generational time, those kind of things, not as important to be learning those equations. It's good to be familiar with them, but it's not really what we're focused on here. We're mostly focused on being able to do this CFU per milliliter calculation. So let's see. The last thing I wanted to go over, they're going to show bacterial growth phases. So I wanted to hit on the 4 bacterial phases of growth. And I apologize, I did not set up a graph here previously. But when you're going to be looking at growth, there's these 4 phases. There's the lag phase, exponential phase, stationary phase, and the decline phase. So when bacteria grow, they kind of look something like this. There's a lag, there's exponential growth, where they're multiplying by 2s because they're going through binary fission. There's going to be a stationary phase where they stop growing, and then eventually there will be a decline. So this is kind of what a bacterial growth phases or bacterial growth curve looks like in an actual experimental setting. So this could be at 24 hours. This could be at 48 hours. So you could get a lag phase and doesn't really look like it's growing. At 48 hours, you could see significant growth. At 72 hours, you could see a stationary phase where nothing's really changing. And then at 96 hours, or whenever your culture is getting really old, you can see that decline in number of living bacteria. So if you, and you're going to be doing this in the simulation, if you plated, so if you had a tube of bacteria and you plated it at 24 hours, at 48 hours, at 72 hours, and at 96 hours, at 24 hours, you might not need to do a serial dilution in order to count what's in there. At 48 hours, you probably will need to do a serial dilution to count the number of bacteria that's in that colony. 72 hours, same thing, because there's still probably going to be a lot of bacteria in there. And at 96 hours, if you start to see that reduction in bacteria, you can tell that you've gone into that decline phase, a rebacterial colony. So just wanted to hit on those 4 phases so that you understand those going into this simulation. I think that was all I really wanted to touch on. Sorry that took so long, but definitely wanted to take some time to answer people's questions about any of this information that we just went through,